Good day to everyone joining us and welcome to today's X Talks webinar. Today's talk is titled Better Target Validation for Drug Discovery with CRISPR-Cas9. My name is Nima and I'll be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for online streaming. You'll be receiving a link to the recording of this webinar via email in a few business days following this event. And you can also access all recordings you've registered for under your My Profile section of xtalks.com. At this point, I'd like to thank Horizon Discovery, who helped to develop the content for this presentation. Horizon Discovery Group is a world-leading gene editing company that designs and engineers genetically modified cells and then applies them in research and clinical applications that advance human health. Horizon's core capabilities are built around its proprietary translational genomics platform, a highly precise and flexible suite of gene editing tools that are able to alter almost any gene sequence in human or mammalian cell lines. Horizon offers over 23,000 catalog products and related research services, almost all of which are based on the generation and application of cell and animal models that accurately recapitulate the disease-causing genetic anomalies found in diseases like cancer. Horizon's commercial offering has been adopted by 1,600 unique research organizations in over 50 countries, as well as in the company's own R&D pipeline to support a greater understanding of the genetic drivers of disease and the development of molecular, cell, and gene therapies that can be prescribed on a personalized basis. And now, I'd like to introduce our speaker for today's event. Paul Russell joined Horizon in 2012 to work on and lead client target validation projects. For the last few years, Paul has developed and performed Horizon's internal target identification and validation programs using both CRISPR and RNAi-based technologies. Prior to working at Horizon, Paul performed research at the University of Cambridge, where he studied the early BRCA genetics in breast and ovarian cancer with Professor Bruce Ponder. With Professor Ashok Venkataraman, he subsequently studied the role of the spindle assembly checkpoint in determining the outcome from a drug-induced mitotic arrest. During this time, he was an early adopter of full genome siRNA screening to address a number of biological questions. So now, without further ado, I'd like to hand the presentation over to our speaker. Paul, you may begin the presentation whenever you're ready.
Thank you very much. Welcome everyone to Horizon's webinar on better target validation for drug discovery with CRISPR-Cas9. My name is Paul Russell and I'm a senior scientist in our therapeutic R&D team based here in the UK. My role here at Horizon is to perform target ID projects and to develop a target validation pipeline for our internal targets. This pipeline can now be utilized for any of our customers wishing to develop their own target ID portfolio. In the clinic today, as for many cancers, cytotoxics are still the mainstay of chemotherapies. The molecular targeted therapeutics over the last 15 years or so have been concentrated on receptor tyrosine kinases such as EGFR, with drugs such as cetuximab and Gleevec going to the clinic. Most often, these targets have positively acting driver mutations. A review of the cancer genome atlas suggests that the frequently mutated targets that adhere to this paradigm have now largely been mined out by Big Pharma. So we need to generate new validated targets for drug discovery programs. In the rapidly expanding field of immuno-oncology, the recent successes appear to be limited to those tumors that demonstrate a high mutation rate and presumably have a high neoantigen load. Thus, it looks like targeting the immune checkpoint will not provide all the answers, especially in those tumors with a low mutation rate. Cancer genotyping efforts have revealed that the majority of driver mutations are either druggable oncogenes, such as KRAS or MYC, or tumor suppressors, such as p53. Consequently, we find that the approach of targeting synthetic lethal interactions with early driver mutations may yet yield a host of druggable targets for future cancer therapeutics. The concept of synthetic lethality would allow us to drug those dependencies that only exist once a frequently occurring driver mutation is present. To date, the most successful example of this is the PARP inhibition story with olaparib in BRCA-mutated tumors. To this end, we have used CRISPR screening to identify possible synthetic lethal interactions with common genotypes. Initial efforts to find synthetic lethal targets in mammalian systems were performed using RNAi technology. Unfortunately, RNAi technology's significant drawback is that it only achieves partial knockdown. As a result, we see great variability between studies. For example, 3 very well-respected screening labs performed shRNA screens to find HIV host factors. Although each lab found outliers, there were minimal overlap in the data generated by each lab. As CRISPR systems can achieve complete knockout of its target, it's unsurprising that Trevor Hart reported that CRISPR screening was 2 to 3 times more sensitive to finding fitness genes than comparable shRNA screens. Therefore, more robust CRISPR results are far likely more reproducible labs. Therefore, more robust CRISPR results are likely to be far more reproducible between labs and studies. So today, in the next half an hour or so, I'd like to show you our experience in performing CRISPR screens and our capabilities to validate hits in order to develop them into genuine drug discovery targets. Firstly, I'll describe to you our primary screens that we've performed in colon and lung cancer cell lines in an effort to identify synthetic lethal hits. I'll then describe to you our high-throughput technology for validating our primary hits using high-density guide screening. I'll then show you how we've developed Inducible Cas9 lines so we can perform further validation studies. I hope to convince you that we've been able to apply these techniques to identify genuine synthetic lethal hits by using a case study where we've identified MCL1 as an essential gene in the presence of FBXW7 mutation in colon cancer cells. Finally, I'll describe to you some of the other gene editing technologies we can apply at Horizon to validate hits. We started by developing our ability to perform CRISPR screens in a panel of 13 colon cancer cell lines. We then expanded this panel to predominantly lung cancer cell lines. We designed a library of guides that targeted 3,000 interesting genes that are druggable using existing chemistry. Each gene in this library was targeted with 10 guides per gene. Additionally, our library contained guides targeting 45 essential genes and 102 non-essential genes. These guides were used as positive and negative controls, and I'll describe to you in a minute how we use these controls to perform our favored analysis. The library also included 100 non-targeting guides that we use as an additional QC metric. We use pooled screening approach with a single construct that expresses both Cas9 and a single guide. The library contains 32,000 individual guides, was delivered into cells by lentiviral transduction at a functional MOI of around 0.3 in order that each cell only ever receives a single guide. Puromycin selection is performed to remove all the non-infected cells over a 3-day period. The cells are then grown for a minimum of 12 doublings or for 30 days, whichever is shorter. The libraries maintain a coverage of at least 300 at all stages. We harvest cells at the end of the puro selection at the end of the growth phase, and the DNA is extracted in these samples and sent for next-gen sequencing using standard protocols. The guide frequency data at these time points is then compared to the guide frequency of the library as it was infected into the cells. All of our screens are performed in duplicate, and the data undergo rigorous QC protocols. In addition to our colon and lung panel, we also performed our primary screens in Horizon's KRAS and PI3 kinase isogenic models in the HCT116 and DLD backgrounds. Colorectal cancer is the most common cancer. That is the third most common cancer in men and the second most common cancer in women worldwide. More than 1.2 million new cases of colorectal cancer are diagnosed globally, with more than 600,000 related deaths annually. The most common mutations found in colorectal cancer are found in the KRAS, APC, PI3 kinase, FBXW7, and P53 genes. This spectrum of mutations was reflected in the colon cancer line panel that we use. Horizon and its founders have an extensive history of working in the colon cancer arena with KRAS and PI3 kinase mutations. We've shown in the past that the assay conditions can be essential to reveal dependencies on these activating mutations. For example, the PI3 kinase inhibitor GDC0941 is an anti-proliferative in the presence of a PI3 kinase mutation. However, this is only revealed under low serum conditions. No such selectivity is witnessed in full serum conditions. Similarly, KRAS mutant cells are sensitive to MEK inhibitors. This sensitivity is greatly enhanced when the cells are grown in 3D conditions. KRAS mutant cell lines in our colon panel are sensitive to SI-mediated KRAS depletion, but only under low serum conditions. As a result, we performed all of our primary and subsequent validation screens under low serum conditions, as growth in 3D is not viable for a pooled screen approach. With our primary screen analysis, we performed two distinct statistical methods to identify hits from the cell lines that have passed our QC. Firstly, we used MAGeCK, which is a commonly used modified ranking algorithm to identify guides that were lost or reduced over the duration of the screen. We also applied another technique developed in Trevor Hart's laboratory in Texas. BAGEL generates a Bayesian score for any gene by integrating the probabilities for each guide that it is either an essential or non-essential gene. The distribution of essential and non-essential genes were determined by our positive and negative controls for each line. Hart's lab has shown that this approach is significantly better in identifying fitness genes than the MAGeCK approach. We too observed this. However, we used both methodologies to establish a hit list, as we were unaware how CRISPR screens would perform and how the data analysis might bias our dataset. As a result, our hit list was very generous and with very loose criteria. As an example of the data we achieved in our primary screens, here's the BAGEL data for PI3 kinase, PIK3CA. As I briefly described, BAGEL is an integration of the probabilities of the guides targeting any one gene to be essential or non-essential. Consequently, a positive BAGEL score of greater than 3 is a measure that the gene is essential. Conversely, a negative score suggests that the gene is non-essential in that line. Targeting PIK3CA in our primary screen suggests that the gene is essential in all the PIK3CA mutant lines. It is, however, dispensable in some of the wild-type lines. In the DLD parental line that harbors a PIK3CA mutation, CRISPR targeting of the PIK3CA gene suggests that the cells are dependent on the oncogene for growth. However, in our isogenic line, where the mutant allele has been knocked out, this appears to be no longer the case. This suggests to us that we could use this isogenic cell line to model tumors that have become addicted to the PIK3CA mutant proteins. Thus, this provides us with a model for testing our putative PI3 kinase synthetic lethal hits. In our KRAS isogenic model, we also saw that the parental line was dependent on KRAS for growth, and that knocking out the mutant allele removes that dependency on KRAS for growth. Another example of a well-characterized dependency is that of P53 wild-type cells being highly dependent on MDM2 for growth. This dependency can be clearly seen in both our colon and lung primary cells. Along with our screening and our cancer cell line panel, we screened our library across a normal breast epithelial line, MCF10As. Unfortunately, there are no good normal colon or lung lines that are amenable to screening. We use this line as a model of potential toxicity that you might expect to see when targeting any putative hit. As we can see here, the PI3 kinase is not essential in the MCF10A line. And as expected, the P53 normal line, we see dependency on MDM2. Screening in normal lines helps us build confidence in the target as a potential therapeutic. So I'm now going to move on and tell you about our validation screen approach. As I mentioned, we generated hit lists with very loose criteria from the colon line data generated against all the common genotypes. We also included a number of genes that had been reported in the literature as putative synthetic lethal targets. In total, we took 708 genes into our second round of screening. For our validation screens, we took inspiration from observations reported at the VAPOC labs, where they reported that dropout rates were much higher in the regions that were functionally significant. This data would be highly significant for any future drug discovery program, so we decided to go with a high-density tiling approach to designing guides for our validation screen. For 530 of our strongest hits, we designed guides across the gene at approximately one guide to every 5 to 10 amino acids, depending on the size of the protein. This averaged at 120 guides per gene. For the remaining 177 genes, we designed 20 of the best guides that did not overlap with the guides included in the primary screen. We also removed any guides that would have been predicted to target the genome in more than 3 places, although most guides in the library were sequence unique. My colleagues at Horizon had developed the CRISPR technology by improving the tracer RNA. This new development was published in scientific reports. So we included this improved tracer in our validation screen that were otherwise performed in the same way as the primary screen had been. We synthesized the library and screened it across the panel of colon cancer cell lines that had been shown to be amenable to screening and represented the spectrum of mutations that we were interested in. Included in our validation screen were the same control guides that we'd used in the primary screen. Here you can see the improvement in the screen performance between our primary and validation screens. Here we've plotted the average bagel factors achieved across all the cell lines in the validation panel. There's a much clearer distinction between our core essentials and our non-essential controls. The most effective use of this high-density validation screen approach was that we had on average 120 guides targeting a single gene. This use of multiple reagents helped to build confidence in the results. Unfortunately, we didn't observe the functional domain dropout to the extent reported by Shi et al. However, we did observe some interesting results. In this first example, a mitotic kinase appears to have a greater dropout surrounding a key phosphorylation site in the kinase domain. Also, twist neighbor is an example of a PI3 kinase putative hit we identified from an siRNA screen we performed in the same colon panel. Twist neighbor is a component of the RNA polymerase I complex where it binds to rRN3. Although in our CRISPR screen, twist neighbor appears to be essential in all the lines that we tested, the guides targeting the domain that binds the rN3, shown in blue, appears to be more sensitive. Overall, the high-density screen gave us much more robust data. As bagel is an integrative metric, the use of more guides per gene gave us much greater differentials between the essential and non-essential results. For example, in the case of the PI3 kinase, we now see that there is a clear distinction between the PIK3CA mutant and wild-type cells. In the case of guides targeting MDM2, the relationship still holds as expected, but the bagel scores achieved a much greater and give you greater confidence in your result. Of interest was that the individual guide data was very reproducible between the primary and validation screens in each line, probably due to the nature of CRISPR being a more robust system. So we now have a number of hits that come through our validation screen. We knew over the course of the screen that these genes were essential in each line. We now had to develop them into bona fide drug discovery candidates. The validation screen gave us many hits that appeared to have synthetic lethal interactions with either KRAS, PI3 kinase, FBXW7, or P53. We stratified these hits on the number of cell lines that the target was deemed essential in, the mutant lines, and placing an emphasis on how dispensable the gene was in the wild-type lines. In the case of KRAS and PI3 kinase genotypes, we also had isogenic data that added confidence that we were dealing with a gene that was reliant on KRAS or PI3 kinase mutation to generate the dependency. However, the screens only generate a statistical metric on how essential the gene is and gives little insight into the magnitude or timing of the dependency. For this, we needed to go into an arrayed format. As we're mostly searching for genes that are essential in the presence of a mutated genotype, it was necessary to develop an inducible assay that would allow us to gain further insights into the phenotype of targeting our hit in the presence of the genotype of interest, and to do this in the most efficient and cost-effective manner. So we set about creating a target pipeline that would allow us to quickly assess the essentiality of any target for a potential drug discovery program. Here is an example of a putative KRAS hit. In this example, the gene was not a strong hit in the primary screen, but was essential in all the KRAS mutant lines, here shown on the left, and was dispensable in the 2 of the KRAS wild-type lines. In the validation screen, where we have many more guides targeting the gene, we observe that the 3 KRAS mutant lines are sensitive to the knockout of this target, whereas in the wild-type lines it is mostly dispensable. Of interest, the one wild-type line in which the gene is essential is also the one KRAS wild-type line that is sensitive to KRAS knockout. The gene became of great interest as the isogenic cell line data suggests that when you knock out the KRAS mutant allele, this protein becomes dispensable. And this is not seen when they knock out the wild-type allele. To study this gene further, and others like it, we decided to develop a panel of inducible Cas9 lines that we would be able to study the essentiality of any targeting. We would then be able to further understand the magnitude of the response, perform the dreaded cDNA rescue experiments, and perform mode of action experiments. For future drug discovery programs, we would then be able to use these lines to discover biomarkers for the loss of target's function. Finally, we would be able to use these lines in any future xenograft studies to assess whether we saw a reduction in tumor growth when we targeted the protein. To generate these lines, we transduced the cells with a commercially available editor inducible Cas9 construct generated by my colleagues at Darmagol. The Cas9 is under a TET-inducible promoter. The construct also expresses a blastocyte resistance marker. As you can see, in selected wells, we can detect a clear induction of the Cas9 protein after dox treatment, as detected by Cas9 immunofluorescence. Into these cells, we deliver the guide and tracer RNA using another lentivirus construct that also expresses turbo-GFP. This allows us to infect the cells under conditions where we achieve greater than 90% transduction efficiency with the guide construct. An intracyte proliferation assay with these cells expressing guides that target either a neutral, in this case KZN, or essential SF3B gene shows that we can observe the dependency of the essential guide, and hence any of our putative hits. Unfortunately, it's never that easy. For most lines that we've transduced with the inducible Cas9 construct, the blastocyte-selected pools show no or minimal growth effects in the presence of the essential guide after dox treatment. So it was essential to go through a round of clonal selection. After performing single-cell cloning on selected pools of HCT116 cells transduced with our inducible Cas9 construct, we then infected those cells with either the neutral or essential guides. It was noted that only a small percentage of the clones showed very clear dependency in the presence of the essential guide. Further characterization of these clones with IF and Western blotting shows that the most functional clones showed the most homogeneous expression of Cas9 after dox treatment across the clone. This allowed us to screen our clones based on their Cas9 expression prior to transduction with the essential guide. Unfortunately, Cas9 expression alone was not enough to identify functional clones. We discovered that cells that were homogeneous for their Cas9 expression after dox treatment might be leaky. Here in the case of 3A5.49 cells expressing homogeneous levels of Cas9, clone A and C are showing dependency on the essential guide even without dox treatment. We assume this is because some leaky Cas9 expression in the absence of dox that is below the levels of detection by either IF or Western blot. We also observed clones that never showed dependency on the essential guide, despite having good expression of Cas9. Therefore, we concluded that it was essential to perform a functional assay with the essential and neutral guides to identify functionally useful clones. We also aim to identify clones that retain similar growth characteristics to that of the parental line. Once we had identified good functional clones, we were then able to use these target hits with our guides that we screened within our validation screens. Here is Western blot data utilizing inducible Cas9 clones and guides targeting either MDM2 or our KRAS putative synthetic lethal hit. In the H838 line, we can clearly see the induction of Cas9 corresponds with loss of the MDM2 protein 72 hours after dox addition, with all three guides tested. Again, with our KRAS target, we see the majority of the protein is lost at 72 hours post-dox addition, with protein down to almost undetectable levels by 120 hours. Again, we can see in all four guides tested, similar results were achieved. This target was assessed in 5 different inducible Cas9 lines with 3 different guides. All 3 sequence-independent guides gave very similar results. It appears, at least with this protein, that the level of residual protein is dependent on the basal levels of expression prior to Cas9 expression. We have seen similar results in knockdown in all of the half a dozen targets that we have characterized to date. This data suggests that the inducible CRISPR system gives very robust results, although it has to be noted that we have seen clonal variability within a cell line with dependency to different guides. Up till this point, I've described the experiments that we've used guides delivered lentivirally. This requires cloning the guide into the construct and then generation of the virus, all of which takes time and resources. Our inducible Cas9 lines are also effective with the transfection of Horizon synthetic guide RNAs. These crRNAs can be co-transfected with the tracer RNA with standard transfection reagents. Here we transfected the A549 Cas9 clone with guides to our essential gene, PLK1 and MDM2. We then assessed the proliferation after 96 hours after dox addition. You can clearly see an anti-proliferative effect of the guides only in the presence of dox. This methodology could easily be used to triage any hits, as it would be easy to acquire these guides in arrayed format for a small scale screen. You are not limited to short-term assays with the inducible Cas9 lines. We have also used them in longer-term assays that reveal dependencies where multiple cell cycles are required to show their phenotype. Here we have developed a GFP competition assay. The guide is transduced at conditions that achieves approximately 50% transduction efficiency. The resultant cells are maintained in culture after dox addition. Dependency is revealed by the growth disadvantage of the GFP positive cells as measured by FACS analysis. Cells are reported as the percentage of GFP positive cells after correction to the untreated sample. Here we show the classic dependency of SMARCA2 in a SMARCA4 mutant line. The dependency is exaggerated after a period longer than 7 days. We have discovered similar dependencies that only revealed themselves after 7 days. We have also utilized these cell lines in standard colony formation assays. The assays are run over a 2-week period and have revealed dependencies that were not seen in the shorter-term proliferation studies. So I'd like to draw this all together with a case study of a target that we can talk openly about. MCL1 is an anti-apoptotic member of the BCL2 family. we identified MCL1 as being highly essential only in FBXW7 mutants. FBXW7 is known to be a target MCL1 for SCF-mediated degradation. In FBXW7 mutant cell lines, MCL1 has been shown to be elevated, which can lead to a prolonged mitotic arrest. In this slide, I will show a summary of the data we've generated for the MCL1 target. In the primary screen, we can clearly show that MCL was essential in all but one of the FBXW7 mutant lines. The only one of these wild-type lines, and only one of the wild-type lines was it deemed essential. In our validation screen, where we had 49 guides targeting the 350 amino acid protein, 4 of the 5 mutant lines MCL1 was essential. Again, as in the primary screen, MCL1 is dispensable in the SNP5 cell line, whereas only one of the wild-type lines show dependency on MCL1. Those guides that target the BH domains in MCL1, which regulate its activity, are among the most sensitive guides in these dependent lines. In 2 of our inducible Cas9 lines that are mutant for FBXW7, we can see an anti-proliferative effect in the presence of the MCL1 guides. In these experiments, we target 3 functional clones with 3 guides that target the gene of interest. We have witnessed some clonal variation within the proliferation analysis, so it's essential to run multiple clones. The dependency on MCL1 is not seen in the FBXW7 wild-type lines. To date, the guide behavior witnessed in the validation screen has been replicated in our Cas9 lines. In our FBXW7 wild-type HCT116 inducible Cas9 line, you can see loss of MCL1 with all 3 guides, but as expected, we do not see any effect on growth. This MCL1 data combined suggests that the compound targeting MCL1 would be most effective in those cells harboring an FBXW7 mutation. And indeed, that is what we see. Servier have an MCL1 inhibitor, S63845, which is undergoing early clinical trials. We use this as a tool compound to test whether our CRISPR data would predict the outcome of treating the colon cells with the MCL1 inhibitor. As you can see here, it appears that from our data that only FBXW7 mutant lines respond to the drug. We have also performed this analysis with a tool compound that targets our putative KRAS synthetic lethal hit. We observed that, as predicted from our CRISPR data, that a compound that targets our KRAS hit was much more effective in KRAS mutant lines. So far, our CRISPR validation pipeline has identified synthetic lethal interactions that is capable of predicting the efficacy of pre-existing compounds. It thus follows that the hits we identify will make good drug discovery targets for those cells which contain the predefined mutational biomarker. In the final part of the webinar, I'd like to tell you about other resources that we have to validate targets. Horizon Discovery is renowned for its precision gene editing. This is obviously a way to generate many tools for target validation, but it is still a relatively slow and labor-intensive process that we can apply once we are very confident in our target. Firstly, we developed a Degron system that we've used for CRISPR targeting the proteins. The nuclease dead Cas9, dCas9, binds the promoter start site and can repress gene expression. In our Degron system, the dCas9 is tagged with a DHFR domain, which targets the protein for rapid proteasomal degradation. On the addition of TMP, the protein is stabilized, no longer degraded, and we achieve expression of dCas9. Interestingly, in the absence of TMP, we see a cytoplasmic signal with IF of Cas9. On addition of TMP, we see the expected elevated levels of dCas9, which appears to relocate to the nucleus. In this liver cell line, we see that 2 guides targeting PLK1 reduce proliferation of the cells. and also reduce PLK mRNA levels in the presence of TMP. Presumably, the cytoplasmic form in the protein detected in the absence of TMP is non-functional. This Degron system is viable for the CRISPR-I approach, but could also be used after tagging any target proteins with the DHFR domain. HAP1 is a human near-diploid cell line derived from the CML cell line KBM7. These haploid cells only have a single copy of almost every human chromosome. Consequently, they're an ideal line to look at the effects of knockout across the genome. As you can see, targeting the protein completely removes the protein. We have performed a number of CRISPR screens in the HAP1 lines. One such screen was looking for genes that conferred paclitaxel sensitivity once it had been knocked out with CRISPR. We were then able to use any of the 3,000 off-the-shelf HAP1 lines that we had, a single knockout, to validate our findings. In our HAP screen, 4 kinesins had been identified as sensitizing the cells to paclitaxel. Using the appropriate HAP1 lines, we were able to confirm the knockout did sensitize the cells to the spindle poison. Although this sensitization was not by much, it was confirmed in a 1-day clonogenic assay in most cases. The screen also identified the bromodomain containing protein BRD8. HAP1 cells containing this knockout were also more sensitive to paclitaxel in both assays. This confirmed the observation of Yamada and Rao, who identified the sRNA mediated depletion of BRD8 sensitizes colon cells to paclitaxel exposure. Thus, HAP1s can be a very useful and fast validation tool in studying genes that are otherwise non-essential. In summary, there are now an abundance of CRISPR data that has been placed in the public domain. This data can be very useful as a starting point, but any conclusions need to be carefully validated. The primary screens give no insight into the magnitude after gene knockout, and we've seen the large libraries used only have a low number of guides per gene and can result in misleading data. This is probably why primary screen data from the Broad and Sanger labs are not as concordant as I would have expected. And finally, the very complex genotypes of cancer cell lines and the conditions that the screens are performed in will mean that many target genotype associations will be misleading. Target validation by its very nature is a very labor-intensive, multi-step process, setting about building confidence in your target. But it will save you time and cost over the course of any drug discovery process. In our opinion, high-density validation screens are a useful tool to validate or exclude a large number of screen hits at the same time. The inducible Cas9 lines are very labor-intensive to generate, but once you have, they are a very useful tool in characterizing the hits to the level of validating a drug target. And as we have seen, our CRISPR results have been able to predict the outcome of drug treatment across a cell panel and will hopefully prove a rich source of new drug targets. And I will stop there and acknowledge the people who took part in these studies, and thank you for your attention and open up for any questions.
Well, thank you very much for your insightful presentation. Up next is our live and interactive Q&A session. But before we move forward with the Q&A, we have a poll question for our audience. Now, that poll question has been launched on your screen, and I encourage all audience members to vote on this question in real time, and we'll take a look at the results. Now, this question is, would you consider outsourcing your screening experiments? Now, our options here are yes, no, I can do this in-house, or no, not relevant. So once again, I encourage all audience members to vote in real time, and we'll take a look at the results. All right, perfect. Now it looks like most audience members have voted. Let's take a look at these results here. All right, so it looks like the majority of audience members, 57%, have said yes, 14% have said no, I can do this in-house, and 29% have said no, not relevant. And now it's time for our live and interactive Q&A session. I would like to invite our audience members to continue sending in their questions or comments using the questions window for the Q&A portion of this webinar. I've already received a few, so we'll start with those. So our first question here from the audience is, you mentioned that you performed an sRNA screen in parallel or prior to your CRISPR screens. What was the overlap in data generated with the two different technologies?
Yeah. So yes, we performed an sRNA screen in parallel across the 12 cell line colon panel that we used in our primary screens with the Dharmacon smart pools. From memory, the library targeted about 2,300 genes. We identified about 50 potential hits, with twist neighbor being one of them, which is the one I showed. We then designed a CRISPR library against those 50 targets, which contained around about 5,000 guides, so that's about 100 per gene, and put this across our colon panel. The overall result was that most of the 50 sRNA hits were essential in all the cell lines and showed no selectivity for mutation status. We put this down to either the fact that it was complete level of knockout achieved with the CRISPR guides or the actual length of the screen taken. Because the CRISPR screen was done over 3 weeks at least, and the sRNA screen was done over 96 hours. We'd be in a position to revisit this with the Cas9 if we thought any of the targets were worth progressing. But the concern would be the levels of toxicity that we might expect when drugging any of these targets in normal cells. I hope that answers your question.
Thank you very much for that answer. Our next question here is, which design algorithm did you use? And do you blast your guidelines before they go into the library?
Yeah, the libraries we used were designed with benchling, which uses a modified dense algorithm, if I remember correctly. We discovered with one of our early screens where we performed where the guides weren't blasted during the design, that we got some guides that were predicted to cut the genome hundreds of times. These frequently occurred in functional domains that were highly conserved between family members. These guides gave incredible dropouts in our screens, presumably because they've shredded the genome and induced a massive DNA damage response. So subsequently, all of our libraries go through a blast step. I'm not sure whether or not the latest benchling algorithms incorporate a blast step into their design. We've moved our library design over into Dharmacon. Their edit R algorithms that they use appear to give us guides with much better performance.
Thank you for that answer. Our next question here is, how many of your primary hits validated in your secondary screens?
Well, that's a very difficult question really to answer, because there are no absolutes with the hit calling, because you're talking about building confidence. We did observe very good concordance with the guide behavior between the primary and validation screens. And as I said, our screen hit list was very generous. Mostly speaking, if the guide showed a strong phenotype, then individual cell lines results, whether or not they were dependent, would be repeated. The validation screens gave so much stronger data than we mostly followed these hits, even if the primary screen data had been weak, as in the case with our KRAS synthetic lethal hit. So it's very hard to actually put numbers on these.
All right, thank you for that answer. Now we have another question here, and this question is, how many clones did you have to screen to get good functional clones?
A lot. This was a very cell line specific. We mostly functionally screened 96 Cas9 positive clones with the essential guide. In cell lines such as the HCT116s and HT115s, we managed to identify functional clones, which were about 20% of all the clones we screened. In other lines, that rate dropped to 1% of some cell lines. Generation of these clones requires an awful lot of effort, but I think it will prove worth it going forward.
Well, thank you very much for those answers. We've reached the end of the Q&A portion of this webinar. If your question wasn't attended to during the Q&A session, the team from Horizon Discovery will do their best to follow up with you after the webinar. You can also reach out to our speaker by using the email address that is displayed on your screen, info@horizondiscovery.com. Thank you everyone for participating in today's conference. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our webinars. I also just sent you a link in your chat box where you can share this webinar with your colleagues on LinkedIn. Now, please join us in thanking our speaker by sending a quick message through the chat panel. We hope you found this conference informative. Have a great day, everyone.